Hi all, hope you all are doing good. So today we are going to discuss about liver. So the liver is the second largest organ of the human body. Then which is the largest organ of the human body? It is nothing but our skin. So the skin is the largest organ of the human body and the liver is the second largest organ of the human body. And the liver is the largest internal organ when we compare it to the various internal organs which are present in the human body, like you have your heart, you have your lungs, you have your kidneys. So liver, when compared to all these internal organs, is the largest internal organ. And the liver is also called a gland. Why the liver is called a gland? Because a gland is any organ that makes and releases substances, and liver is an organ which makes and releases bile. So that's why the liver is called a gland. And when compared to all the glands which are present in our body, like you have your thyroid gland, you have your pituitary gland, you have your pancreas, the liver is the largest gland as well in the human body. So, the liver is a vascular organ. Why the liver is called a vascular organ? Because the liver contains a dense network of blood vessels like your arteries, your veins, and your capillaries. So, that's why the liver is a vascular organ. And the liver is no wedge shaped. That is, it is broad at one side, and as it moves to the other side it narrows. So that's why the liver is wedge shaped, and the liver is reddish brown in color, and it weighs about 1.4 kgs to 1.6 kilograms. That is how much weight the liver has. And we'll look into a special feature of the liver. The liver is the only internal organ in the human body that can completely regenerate. That is, it can completely grow back. So, suppose if the liver is cut and only 20 to 25 percentage of its original size remains, from that small 20 to 25 percentage part, the liver can completely regrow its original mass. So, completely it can regenerate into this complete organ with a small piece. So in case the the part of the liver is smaller than 20 percentage, example it's 19 percentage or 18 percentage, very less small, no very small part or less part is remaining, then the liver will not be able to regenerate. So that is one limitation of liver. It can regenerate completely with 20 to 25 percentage of its original size. If it's lesser than that, it cannot completely regenerate. So now we will see where the human liver is actually located. So if we see the liver is located in the upper right part of the abdomen, okay, and it is just below the diaphragm. So diaphragm is a muscle, okay, which is present here, and above the diaphragm you have the lungs are covered by the rib cage, and the lower part of the rib cage, the lower right rib cage, it covers this liver and protects this liver. So this is where our liver is located. So now we will see what are the various functions of liver. The liver can perform nearly 500 plus vital tasks. So many functions of liver are there. So 500 plus tasks the liver does inside our body, but we are going to discuss only few of the important functions of liver in a high level. So one of the function of the liver is metabolism of nutrients. So it helps in the breakdown of the nutrients. So what are these nutrients? You have your carbohydrates, you have your fat, you have your proteins. So whenever you eat food, you have all these nutrients like carbohydrates, fats, and protein, which are broken down into usable energy by our liver. So that is one of the functions. The other function of the liver is bile production. So liver produces bile, and this bile is used for the breaking down of fat, and that is what our liver does. So liver produces bile, the bile is used to break down the fat, and the fat is absorbed by the liver. And the bile is greenish yellow in color, which is a fluid generated by the liver cells. Then liver helps in the urea production. So whenever the proteins are broken down by the cells for no uh, doing bodily functions, that generates ammonia, and ammonia is very poisonous. It's not no good for our body. So that ammonia toxin is converted into urea by our liver, and it is sent or excreted via the kidney. So the blood is urea is sent through the blood to the kidneys, and kidneys excrete it via the urine. And the liver helps in the production of albumin, which is a plasma protein produced by our liver. It produces that plasma protein. And what is the use of the albumin? Albumin is a special protein which is produced by our liver, which is used for the transportation of the molecules in your blood vessels. So in your blood circulation, if the molecules have to be passed, or the hormones or any other components or substances needs to be transported in the blood vessels, they require transportation vehicles. So albumin is your transportation vehicle. This is a protein which is produced by our liver. If liver was not producing albumin, what will happen? It will lead to the non-transportation of molecules. The molecules will not be transported, and also albumin helps in maintaining osmotic pressure. So what osmotic pressure does inside the blood vessels, if this osmotic pressure was not maintained inside the blood vessels, what will happen? The water which is present inside our blood vessels will leak out, and it will lead to the dehydration. So it maintains the fluid balance inside our blood vessels because of this albumin protein. And the liver helps in storing of the vitamins and minerals like A, D, E, K, B12 vitamins, and minerals like copper and iron are stored by the liver. And liver helps in our immune system support. It has special cells which help in removing the bacteria, viruses, and other pathogens from the blood. So it helps us in maintaining the immunity of the body. And it helps in hormone regulation. So how it does the hormone regulation? If there are any excess hormones present in our blood, like if you have your more insulin is there, or you have more estrogen, it breaks down and clears those hormones. That's how the liver does hormone regulation. And then the liver helps in the blood filtration. How the blood filtration is done by the liver? It removes all the toxins from our body, be it any chemicals or your medicines which you are having, any foreign particles which enter your blood. The liver converts those toxins into non-toxic form, or it neutralizes them, it attaches some molecule to them so that they become non-toxic, and it excretes those toxins via the bloodstream or via the bile. So that's how the liver does the blood filtration work. And the liver helps in blood clotting as well. The liver generates proteins called fibrinogens, which help in the clotting of blood. So wherever the injury site is there, these fibrinogens go and act like the internal band-aids. So what happens? The excess bleeding will be stopped. In case the liver was not producing this fibrinogen proteins, even a small cut will no bleed more. So that's how the liver helps us in blood clot. And then the liver helps in a blood sugar regulation. How does the liver help in blood sugar regulation? It converts the excess sugar which is present in the blood, it absorbs it and converts into glycogen and stores it in the liver. So whenever our body requires sugar, or our glucose level drops, or it requires no sugar, the liver will release that stored no glucose to the bloodstream. That's how the blood sugar is regulated by the liver. So these are few of the primary functions of the liver, and there are many other functions which the liver does. So see how much important the liver is for our body. Now, since we have seen the different functions of liver, now let us get into the anatomy of liver, how the liver looks, what are the different parts in the liver. That is what we are going to see now. Now, if you see, this is the liver which looks like this when we see from the front. From the front side, when you look at the liver, it's somewhat looks like this. It is divided into two big lobes, no, right lobe and your left lobe. Okay, we have two more lobes. Totally four lobes are there, but these are the major lobes which are present in the liver: the right lobe and the left lobe. These are the two main functional lobes present in the liver, and then these are no partitioned. A visible partition is present, right? This is nothing but your ligament. So ligament is one which helps to place the organ or keep the organ in place. So this is nothing but your falciform ligament. So this shows us a visible partition between the right lobe and the left lobe. And you have your inferior vena cava, which is a vein from which you have the hepatic veins entering your liver. So what are these hepatic veins? The hepatic vein is nothing but hepatic means something related to liver, like renal is related to kidney, pulmonary related to lungs. Similarly, hepatic is related to liver. So hepatic vein, the vein which enters the liver is your hepatic vein. Then you have your aorta, which is your largest artery. From this aorta, if you see down, the blood supply is given to the liver, which is hepatic artery. One third of blood supply is sent to the hepatic artery to the liver, and it supplies nearly one third of blood supply is no received from this hepatic artery by the liver. And hepatic artery sends you the oxygenated blood, and the remaining two-third parts of the blood supply which the liver no needs to get is got from hepatic portal vein. So this supplies the remaining two-third of blood, the hepatic portal vein. So what is this hepatic portal vein? Is this portal vein, if you see, this gets the blood supply, all the venous blood draining drainage from all the digestive organs is drained into this hepatic portal vein. So I will show you that so that you understand. So now this is your liver, and these are all your digestive tract organs. You have your intestines, large and small, you have your stomach, you have your spleen, you have your pancreas. So all these organs no drain their venous blood, the deoxygenated blood, into this hepatic portal vein which enters your liver. So instead of no releasing that blood into no inferior vena cava vein, it releases that blood into the liver's hepatic portal vein. So two-thirds of the blood supply of the liver is received from all these organs, venous blood. So that is what is your hepatic portal vein. Hope you understood this. Next is your no bile duct. So bile duct, this is your gallbladder. This temporarily stores your bile which is generated by your liver. So the liver generates bile. It no goes via this duct which is your hepatic duct, enters this common bile duct, this common bile duct, and from the gallbladder it enters the common bile duct via this cystic duct. So this channel or this tube is called your cystic duct, and this is your gallbladder. So this common bile ducts later goes and connects to the small intestine. So this bile is released into the small intestine via this common bile duct. And now you have this another ligament called the teres ligament. So this is, there is a round structure which is present uh, at the bottom, that is your teres ligament. Okay, I will tell you how this teres ligament and the falciform ligaments are formed. So first we will see what is covering the liver. So just understand these lobes which are present in the liver are like no acting as the independent functional units. So they have their own blood supply, they have their own venous drainage system, independent. So all these segments act independently. These lobes are further divided, no, they into 8 segments, and they all no act as independent lobes or segments. So basically these lobes are independent units. That's what you have to understand. And then this entire liver is there, right? It is covered by 2 membranes. Okay, the first innermost membrane which covers the liver is your Gleason's capsule. Okay, that is very tough and fibrous. The Gleason's capsule, that is the membrane which is covering the innermost layer, innermost liver tissue, the soft liver tissue, which is protected by this Gleason's capsule, which is very tough and fibrous. And covering these Gleason's capsule, on top of this Gleason's capsule, we have what is called your visceral peritoneum, which is very smooth and slippery. It wraps around the entire Gleason's capsule layer. Okay, so now this visceral peritoneum is what is folding together here. So it is like the plastic is wrapped on top of the liver, and here no, there is no the space between this left lobe and right lobe is there, right? So that here the visceral peritoneum folds, double folds, and it is attached to the liver, and it forms this ligament. So that is how this ligament is formed. This ligament is formed from the visceral peritoneum, that is the outermost layer, okay, that wraps around the outside of Gleason's capsule. So this one only folds and no, forms this visceral falciform ligament. So that is how this ligament is formed. So now that you have seen this, we will see this teres ligament in the, because this is front part only, less is visible, back you will be able to see properly the teres ligament. So now if this liver is turned backside, no, the other side of the liver, now you are seeing the front of the liver, now we'll see the back of the liver, how it looks. Okay, so this is how the back side of the liver looks. So if you see back side of the liver, now this is your teres ligament which I was telling in the front view, which is visible only somewhat in the front. So this is your teres ligament, which is also called the round ligament. Okay, what actually is this ligamentum teres is, this basically it was an umbilical vein, no, which was entering into our liver when we were in fetal stage. So when we were a fetus, from your mother's body, your umbilical vein was entering our liver. So that is what is this ligamental teres that got dried up and it became something called ligament teres. So this is the ligament, and the next ligament is your ligamental venosum. So how this was formed? So there was another no ligament, or we can call the blood enter, no, via this, it bypassed this section. So 70 to 80% the blood was entering the liver, and this particular blood vessel bypassed the liver and it directly drained into inferior vena cava. So that got converted to what we call as ligamentum venosum. So in the fetal stage, this blood vessel which was bypassing the liver and draining directly into inferior vena cava got dried up and it became ligamentum venosum. So these two are that ligaments which have been formed during no, our fetal stage. These two blood vessels were, dried up and formed ligaments like this in our liver. And this part in between, this is the porta hepatis. What is porta hepatis? It's nothing but the hilum or is the opening from where all your arteries, veins, your bile duct enter the liver. That opening is called your porta hepatis, the fissure or the small opening or tear which is present in the liver, to which all these vessels or the ducts enter the liver. That is porta hepatis. And now if you see, you have two more lobes. I was telling that the liver has four lobes. We saw the right lobe and left lobe from the front, and these the other two lobes are small lobes which are present at the back of the liver. So the upper lobe is the quadrate lobe, which is present between the inferior vena cava and the ligamentum venosum. Within this ligament and this inferior vena cava, we have the quadrate lobe. See, if you see here, it is having this below some W shaped shape as well. And the below one is your quadrate lobe. Quadrate lobe is the lobe which is situated between the gallbladder and your ligamentum teres. That is this ligament and this gallbladder. That lobe is your quadrate lobe. So you have totally four lobes: left lobe, right lobe, quadrate lobe, and quadrate lobe. Because no, these both are no very small in size. That's why uh, they are not visible in the front, because both are present at the back. So that is the back side of the liver. Now we will see, if we take a small part of this liver tissue and we will try to see what is present in this liver tissue. What are those microscopic structures which are present inside the liver? Like we have microscopic structures in kidneys, we have in lungs. So in liver, what are those microscopic structures? So if one small part of tissue of the liver is taken and it is seen under the microscope, you will see something like this. You'll have a honeycomb like structure with many hexagonal lobules present. So hexagonal structures are present, forming a honeycomb structure. So all these hexagonal structures are called your lobules. So these are the microscopic structures which are present in the liver. So we have the hexagonal structures called as lobules, and we have honeycomb structures, a group of lobules, no, arranged in the form of honeycomb structures. So throughout your liver, if you see, right, you will have your these lobules present, and they are all arranged like this. We have the entire liver made up of the lobules. So lobules are stacked one no, upon each other or at the sides. So that's how the entire liver is formed. And each lobule, if you see, has one central vein in which the blood drains, and you have your portal triad. What is portal triad? It is the three portals. One is arteries, veins, and your bile duct. So three together constitute the portal triad, and this is your lobule, individual lobule. So now we will look into the individual lobule, how individual lobule looks. So this is how your single hepatic lobule looks. Now the size is only 1 to 2.5 mm size. So that much small the single hepatic lobule is. And if you see here, it's your central vein. These are your portal triads, your artery vein, and your bile duct. And these are your sinusoid which are present. So you'll see what is a sinusoid in detail. These are no special capillaries which are present in the hepatic lobules, special type of capillaries. We'll see that in a while. So if you see, it is having hexagonal structure, it has 6 sides, 1 2 3 4 5 6 sides. And this is how your single hepatic lobule looks. Now we will look into the sinusoids. Now if you further no zoom it and see, you will see this is one section of your lobule. Okay, so this is your central vein where your blood drains into the no uh, hepatic vein, and then it goes into the systemic circulation. And if you see here, these are all the sinusoids. I was telling that right, there are special capillaries present. So these pink color ones, whatever you are seeing, the pink and this light blue one, this all are your sinusoids present in your lobule. So these sinusoids all drain the blood into central vein. And here is your portal triad which we saw. We have the branch from the hepatic artery, we have the branch from the portal vein, we have the bile duct. So all these three together constitutes the portal triad. Three, tri means three, triad. So portals. So different portals are present. And these are the sinusoids. So if you see, these sinusoids are special capillaries. Understand, they are capillaries. So capillaries are there, but they are also one form of blood vessels. So we will see in detail how the sinusoids look. But understand, they are special capillaries which are present in the lobules. Multiple sinusoids are present. They are all interconnected, forming a web-like structure. So what happens? Basically, they help in mixing the blood. We know the blood is received from artery as well as portal veins. So oxygenated and deoxygenated blood. So we know from artery we get oxygenated and vein we get the deoxygenated blood. So these sinusoids help in mixing of that blood, and the sinusoids also help in filtering the blood, removing toxins, observing important nutrients from the blood, storing okay different storing of the important nutrients, all is done when the blood passes via these sinusoids. So sinusoids are channels. When they pass through these lobule, the blood is cleaned, nutrients are observed, sent back into the bloodstream, and finally the blood is sent or drained to the central vein. And if you see, these lobule is made up of lot of cells. You can see here between the cells these sinusoids are present. These cells are nothing but your hepatocyte cells which are present. So all these cells are your hepatocyte cells which are nothing but your liver cells which do the actual work. So they all the functions of the liver are done by these cells, and sinusoids are the channels which provide the blood supply to these cells. And if you see here, you have different cells present inside these sinusoids called the Kupffer cells, which are macrophage cells which eat up bacteria, virus, or any pathogens. and there is space between these hepatocyte cells and these sinusoids. These sinusoids and these hepatocyte cells, we have a gap in between them, and that gap is called the space of dish, and special cells called stellate cells are present in that gap, which help in storing vitamin A. Okay, and these sinusoids, no, there are multiple branches going from these portals, and the sinusoids are no interconnected, forming web-like structure in this full 3D lobule, and multiple channels are there from which the blood is drained into the central vein. Okay, so blood is drained into this central vein. So now this sinusoid we'll see in detail. We are studying now how these sinusoids actually look in this lobule. That is what we are going to see in detail. So we'll go further inside and zoom the structure, and this is how your sinusoid looks. So this is your sinusoid. This is the innermost layer, which is your sinusoid. So sinusoid is made up of single layer of endothelial cells, single layer, and there are many gaps in between. They are not continuous layer of cells. There are gaps in between, there are holes in between, which are no penetrations in between the cells, so that the blood which is entering the sinusoids can easily no come in contact with these hepatocyte cells. So these are nothing but your hepatocyte cells. So these hepatocyte cells are there, right? So in between the hepatocyte cells, between two adjacent hepatocyte cells, we have something called bile canaliculi. So the bile is produced by these hepatocyte cells, is released via this bile canaliculi. This is a channel in which the hepatocytes produce bile and release that into this bile canaliculi, and from that bile canaliculi, that is sent to the channel which is running parallel to the hepatocyte cells, and it is finally drained into the bile duct. So this is your hepatocyte cells. They have finger-like structures. They are called microvilli, so that they can absorb the nutrients from the blood. So that's why they have many finger-like structures on the side facing the blood. And now we saw you have lot of gaps, so that this can no plasma can directly enter this space. Now there is a space between these hepatocyte cells and your sinusoid, right? So that space is called nothing but space of dis. This space is space of dis, and here whatever plasma is leaking out from the sinusoid enter this gap, and this microvilli comes in contact the hepatocyte cells, observe the nutrients from that plasma, and take whatever is important, release back, which is no useful for the blood, and remove the toxins from the blood. So that is what the hepatocytes do. The filtration work is actually done in the sinusoids. Absorption of nutrients, storage of nutrients, all is done in the hepatocyte cells here. And they have special cells in sinusoids called Kupffer cells. What these cells do? These are the macrophage cells which eat up any bacteria, virus, or pathogens which enter the bloodstream. And we have these stellate cells which are present in the space of dish. These cells help in the storage of vitamin A. Also, they help in blood regulation by contracting and expanding. They change the pressure of the blood flow. And now we have these endothelial cells with lot of gaps in between, so that plasma can enter. The reason why we are discussing about these gaps, so you can ask why we even require this endothelial cells, why the sinusoids no cannot directly flow here no completely, why these are still protected by these endothelial cells and small small gaps are only present. This is present because if the gaps were very big or it was completely open, even the red blood cells will fall into this no gaps and it will lead to internal bleeding. To avoid the bleeding, the red blood cells are large so that they cannot enter these gaps and they remain in the bloodstream without causing internal bleeding. That's why we have small gaps in the endothelial cells and it's not completely open. Now what happens? Now whatever plasma is there from the blood, it enters the space of dish. So these microvilli comes in contact with that space plasma and it absorbs all the important nutrients required, and whichever nutrients need to be sent back into the blood, it sends back to the blood. And if there are any toxins or any other things present, it converts the toxins into forms so that it does not affect our body and sends out excretes those toxins through this bile. So those toxins are excreted via this bile, and via the bile it is excreted into the gallbladder, the bile, and then it connects to the small intestine and it is excreted out of the large intestine. So that's how it removes the toxins. And some toxins which are very small and water soluble, no, they sent it back into the blood. But after converting them or neutralizing those toxins, they sent it into the blood, and the blood enters the kidneys and the kidneys filter those toxins afterwards. So that is how your liver does the various functions. So you can see at the microscopic level how the liver is helping to filter the blood. So what happens now, we'll see what happens now. Here the bile is sent out and here the blood is cleaned and sent via the sinusoid. So we'll go back. So these are the sinusoids which we saw. They are filtering the blood, all it is drained into the central vein. And bile, we saw this bile is drained, generated bile is drained into the bile duct. Now if you see, the bile is always flowing in the opposite direction than the direction of the blood, because if it flows in the same direction, there are chances of mixing of bile with blood which can no harm our body. So that's why the bile always flows in the opposite direction of the blood. The blood is flowing that side, the bile is flowing this side. Okay, and so once the blood is purified and sent there, toxins are sent out of the blood into the bile duct. And if you see, the hepatocyte cells which are facing the bile channel are tightly packed so that the no bile does not escape to the other side into the bloodstream. That's why they are tightly packed, and towards the sinusoids they are loosely packed so that they can absorb lot of blood into the hepatocyte cells for filtering purpose. So that is how the cells are present. The hepatocyte cells are present around the sinusoids as well as the bile channel. So once these are no filtered, what happens, all the from all the sides these sinusoids drain it into central vein, and all these are drained into central veins, all joined together, and then they are sent to the hepatic vein, and from the hepatic vein they are sent or drained into the inferior vena cava, which goes into systemic circulation via the inferior vena cava. That is how the entire process of liver function works. So, hope you have understood the various functions of liver and you have enjoyed today's session. If you like my video, please like, share, and subscribe. Thank you.